Native American names tend to have things like that in them. White feather, pretty places. Yeah.
That's not in a file, it's in.
No, this is this goes in evidence. Yeah, thank you.
Where did it go?
Anyone else? No. Cause it says aside from hope, did anyone else?
It's the name. The chamber guy.
You guys got about 3 minutes, so you should probably pack up a bit.
I can't.
Which evidence do you want?
Wait, why did the dog bark? Smell the dead body?
Yeah.
The struggle and hopefully a little bit of dog.
The dog was not downstairs, so I'm gonna hope someone else.
Yeah, the dog would try to cross the way, and the door was open, right? He stumbled in the door, he didn't close it behind him. Went in the door, and strangled him.
Yeah.
Nobody was up at 61 year old everyone looks. He shouldn't have been up, I would have been calmed down.
So like, we can tell this guy did not do it because he does not.
The dog. The mailman. I'd have to do that.
Okay, well, that's.
This is the interview and
Yes. Ethnicity must be great. Yeah, there's not a lot of.
don't forget the.
Yes. You know, to be older than that, we just don't know. Yeah.
Yeah. There's a little here.
Just like. Look like I. Oh, no. Yeah, no, that you talk to me. I think Brian, I don't like this just.
Gotta get it done right.
Does Brian have a?
And you have to do it by yourself. That's what we went home and did, took all of our stuff.
Love it. I do love it. I love all the hard work. People working hard. Yeah. Working hard.
From the.
That's very true.
Say the timeline,
No, you just did that like in, yeah, just like now. Yep, that's perfect. Thank you.
You're on base.
and same for.
Yeah, those can stay out too. That is perfect. Thank you. You guys are so organized. That's exactly what I wanted. Outstanding.
Is this one on?
But I'll get you good. Right. Junior, see you next week. Come on, Gansy. You hang on that. Just tuck it in your folder.
I love your new haircut.
It does, doesn't it? Thank you. Look at this. Woohoo! We got one solved, returned, and they have a press release, which is what your goal is, that press release. That means you're announcing to the world that you've got your guy.
Is this one of those?
Maybe Emerson?
This yours?
There you go.
There you go.
Yes.
No.
All right. Let's get rolling, people. Are we having fun yet? We've got one solve. We've got 1 minutes away from a solve. I anticipate at least a third next week. So we're getting there. How many what?
Yeah.
Well, the thing is where they're 9, but where you are, once you get into the gene tree database, that's all work you can do independently. So like from this point on, you've entered your NATIS, you didn't get a hit. You applied for your GenPick profile, you got your GenPick profile. Then you went ahead and said, yes, please let me into gene tree. Then it let you into gene tree. Now you got to build your trees. And then once you have that, you can come and tell me, we've built the tree, but you have to do this in person, give me the form. We built the tree and now we need to request a direct DNA comparison. That's your next phase. When you get that direct DNA comparison, once your tree is built, for those of you who are working on trees, once you get that, it's going to let you into another digital part of the lab, and you will just suddenly be confronted with your suspect, and you'll have to deal with him or her. Well, him, you know it's a him. Everybody knows it's him at this point because everybody has seen that the profile has an XY in it. Okay, so we will roll through this, and maybe there'll be a little bit of time at the end of class. I kind of doubt it, but if we go quickly, maybe. And then those of you who are working can use that little bit of extra time. So this is our last lesson of Unit 4, which means you're going to do what this week? Get a test. All right. Oh my gosh, stop calling me. So yeah, after we left class last week, James Watson died. If you didn't catch the news, it was all over the news and all over social media. So one of our 4 people in our story of how the structure of DNA was discovered, Rosalind Franklin had the X-ray crystallography. Maurice Wilkins stole the image, gave it to James Watson and Francis Crick. He was the last surviving of that group of 4. He was 97. So
You seem like everything.
he ended up later in his life becoming an even more controversial person than, I mean, at the beginning, he was just heralded as a hero, right? Because he and Crick discovered the structure of DNA, and that was like a big deal, and that was amazing, and he's a hero, and yay, hooray. And then we find out that maybe they weren't the first people to figure it out, and maybe they did use some of her research, and maybe that wasn't so great. And then later in his life, like up until 2007, he was still denying Franklin's contribution in his TED Talk, which was real fun. And then he made some wackadoodle comments about intelligence and genetic intelligence and race towards the last couple years of his life, which got him actually booted from an organization that he was like a figurehead for. So yeah, he kind of, over time, he kind of tainted his legacy, which is really sad, but he's still part of our story of how we got to where we are today. So no human is perfect, right? Like when we do chemistry and we talk about the scientists who were involved in all of the nuclear chemistry stuff, those are some very complex stories because those guys, a lot of them like fled the Nazis, some of them worked for the Nazis. Like it becomes very gray areas when you get into there. Like there was the one who didn't leave because he thought the Nazi thing was going to blow over real quick. Wasn't that Heisenberg? Heisenberg was like, this isn't going to last. I'll just keep my head down and keep working in my lab. And then that's not what ended up happening. So yeah, history isn't pretty. Okay, so genetic engineering. Here's a wackadoodle thing for you. This here is a Turkish Angola cat. And normally when you put them under UV light or black light, they kind of are greenish. But they've put a gene in them. In some cases in labs, they've been able to put a gene into them that causes them to fluoresce bright red in the dark. That's not a light. The cat is glowing. That cat's doing that. Isn't that fun? Yeah. So, yeah. So, yeah. We do some weird things. How much does it cost? More than you got, I'm sure. Now, when you see stuff like this, you look at it and you're like, are they doing that just for fun? Like, why in God's name would you make a glow-in-the-dark cat? And really, it's more along the lines of, is this something we can do so that we can use it practically? That's usually how these things start. Like in a few minutes, I'm going to show you a glow-in-the-dark tobacco plant. There's no practical purpose for that, but we were able to do it. So that's kind of the point. So what's selective breeding? Can somebody tell me what selective breeding is? Caitlin.
Through selective breeding, humans choose organisms with one to characteristics to produce the next generation.
Yeah, so what we're doing is we're breeding organisms with desired characteristics to produce the next generation. We have done this since we stopped being hunter-gatherers as a species. This is what we've done. And then examples would be things like horses, cows, pigs, pretty much every plant you eat. There is no plant you're going to get, even the little organic ones or whatever that you go to Sprouts to get. They've all been bred selectively to get to where they are. If you've ever seen like wild crab apples and tried to compare that to a real apple, like an apple you're used to, like a real grocery store apple, not that they're not real, they are. But you know what I mean? You see how completely different they are. So we're going to talk a lot about dogs today because everybody's familiar with dogs, and it's a great example of how selective breeding works. So we've been breeding dogs for thousands of years to produce the characteristics we want. And some of those characteristics have to do with how they look, others are their function, others are their particular behaviors or how strong certain instincts are. And it allows those animals with wanted characteristics to produce the next generation. And we use selective breeding, which takes advantage of naturally occurring genetic variation to pass those wanted traits to the next generation. And we've produced new varieties of cultivated plants this way. All domestic animals, horses, cats, cows, dogs, all of these things have been bred selectively for particular traits. Native Americans bred this particular little plant, teosinte, I think is how they said it, but I could be wrong. And that's how we got corn. Now, you probably know from even like when you were little and you read your little board books about Thanksgiving, like the Native Americans had corn. The corn that they had was not the corn they started with. If you've ever seen what it looks like as the wild type, it's this little, it's like maybe this big, and it's maybe as big as your pinky, and it's got like 4 or 5 little kernels on it, but they're little kernels. It's not anything you would want to eat. But they found a way to make it more productive, more nutritious, more bulk, so you get more volume out of it, more food. So another example would be colored cauliflower. Who's seen the colored cauliflower? Have you ever seen it? It's really cool. They usually have it at the funky stores, like Sprouts or Trader Joe's or something. You might see the colored cauliflower. They're not going to have it too often at Walmart, though I have seen it there, the orange stuff there. So how are they doing that? Did they dye it? Did they like dunk it in dye before they put it in the store? No. They bred it selectively to produce these wild colors. And not only are they more attractive and they have a better appeal in the market, but they can have higher levels of vitamins than your regular white cauliflower. So if I'm in the store and I see purple cauliflower and the white cauliflower, I'm going to try that purple. That's pretty cool. So I'm more likely to buy it. So now as a consumer, I'm helping whoever produced that make money. So it's for obviously for financial gain, but also because the product can be better in the end. And then we have selectively bred over 171 breeds of dog that are recognized by the AKC. What's AKC stand for? Does anyone know? Do you know?
Say American Kennel Club?
Yes, American Kennel Club. Now, there's not just AKC, there's also CKC, which is Continental, I think it is. And that covers Canada and the US, like Aussie's CKC registered. But if you have a purebred dog that is registered with AKC or CKC, and then other countries have their other registries as well, but you have to have the pedigree for that dog. And we have been able to breed them into 7 groups. For some reason, I only printed 6 here. The last would be toys. So we have bred them and specified their function or their look, and we've been able to divide that up into 7 groups. So how well do we know our dogs? This is going to be fun. All right, now, this first one is maybe not the best example. I probably should have put that first one in toys because I think technically it's a toy, but it came from this group. So does anybody know what these are? Do you know what they are, Lucas?
One's, I think it's a certain type of terrier. I don't remember what it's called. And then the second one's a boxer.
A boxer and a terrier. What do you think, Barrett?
I think the first one's either a Maltese or a Yorkie. I think it's a small Maltese or a Yorkie.
It's a Yorkie. Okay. Now, what's the second one? Do you know?
Yorkshire Terrier, Pitbull.
Yorkshire Terrier and Pitbull. Now, Pitbull's what we commonly call them. Do you know what they're actually called? It's an American Staffordshire Terrier. So what group are these in? Terriers. These are terriers. What do terriers do? Why do we have terriers?
They're called terriers because they're terrors.
They're terrors. They are go-to-ground dogs, which means they are supposed to get vermin out of the ground. Rats, mice, rabbits, things like that. Yes, they are go-to-ground dogs. Typically speaking, that is what a terrier does. And these are some of the terriers you may or may not be familiar with. So we have lots of little terriers here. So Norfolk and Norwich, I love. They're identical, but Norfolk's ears go like this and Norwich goes like that. Otherwise, they're the same. All right. How about these 2? The one on the right is far easier than the one on the left. Do you know what the one on the right is, Xander? Or either of them?
Great Dane, I think that's I forgot what name I had. I had a name.
You had it and it went out? Do you know, Lucas?
One on Great Dane and the one on the left is a Saint Bernard, I think.
What did you say? A what and a Saint Bernard?
Saint Bernard, I think.
No, it's not a Saint Bernard. Do you know?
Is the one on the left King Charles, and on the right Weimaraner?
Weimaraner. We got a Weimaraner on the right. The one on the left is not a Cavalier King Charles. It's actually a Clumber Spaniel. Aren't they great? Look at that thing. Look at his face. You just want to squeeze our little lips. Mushy face dog. So what group do you suppose Clumber, Spaniel, and Weimaraner are in. The Spaniel is a clue. Do you know?
I'm gonna say herding.
Not
Or-
hurting. Good guess, but not hurting. Do you know, Isabella?
Similar to the bloodhound, so.
Something similar? So those are hounds. That's a good guess. Do you know? So it is a type of hunting. So the group that we have for these are called sporting. These are typically your bird dogs, right? A lot of these are bird dogs. So you're gonna find English Springer Spaniel, Cocker Spaniel. You're gonna find let's see, pointers, those type of dogs, golden retrievers, Labrador retrievers. These are your bird hunting dogs or your sporting dogs. And you go out and you take your bird shot and your dog, and you go out into the woods and you come home with some ducks or pheasants or whatever. That's the purpose of a lot of these guys. Okay. What do we have here? If you don't get the first one, you have to leave my house and never come back. What do we have, Ridley?
Is it a dachshund?
Yep.
And a.
That one's trickier, much trickier.
No, I have the name, it's
Hang on, give her a second. She's thinking.
Scottish Wolfhound?
Okay, you're yeah, very close, very close. So it's a Scottish Deerhound, but yes. But what's the other one that looks just like it?
Irish Wolfhound.
Irish Wolfhound. Okay, so this is your second shortest and your second tallest. Your second shortest is a little dachshund. Oh, a little short thing with a little tiny one. And then you get a Scottish Deerhound. The tallest dog is an Irish Wolfhound, which head is like standing. Their heads are like here. They're massive.
I saw one the other week.
Did
They're
you?
so pretty.
Oh, they're beautiful and they're huge, but they look very similar to a Scottish Wolfhound. So they're big dogs. So I picked your second shortest, your second tallest. What do you suppose your shortest dog breed is, by the way? Do you know?
I think it's
It's hard to be shorter than
Maltese.
No, do you know?
Chihuahua.
Hey, Chihuahua. So Chihuahua is actually shorter than a dachshund if they're well bred. A miniature dachshund, that is. So a miniature dachshund is your second shortest, and your Scottish Deerhound is your second tallest. So what group are these in? Do you know, Drew? Hounds. Okay. These are the hound group. Now, here's the crazy thing. If you look at these hounds, there's not a whole lot of variety in height. They're either really short or really tall. And that's because hounds are further subdivided by a certain characteristic. Does anybody know? Do you know, Drew?
No.
Sight hounds and scent hounds. Okay. So which ones do you suppose are the scent hounds? The short ones or the tall ones? Everybody.
Short one.
Why?
They're close to the ground.
They're smelling the ground, right? And the tall ones are the sight hounds. So like you're gonna find your Afghans, your Borzois. There's your Scottish Deerhound in here. They're tall. I love the otter hounds. They're super cute. And then you get these little shorties like that one. I don't know if you can read it, but that's a Petite Facette Griffon Vendéen, which they call a PBGB. It's so freaking cute, right? They're little short things. Also, you'll notice that a lot of the short ones, the scent hounds, like look at the face on the beagle, on the dachshund. Even look at the face, even though he's tall, on the bloodhound. They have big long ears and lots of wrinkles. Why? What's the purpose of those wrinkles and big long ears, do you suppose? As a scent hound. So as they're moving along, the scent gets trapped in those wrinkles and the big long ears, and it helps them smell where they're going. I follow a couple of Instagram channels for like canine dogs, like police canine dogs, because they're just so stinking cool. I love them. Like I love to watch the fur missiles catch the really bad guys. It's amazing. But I just saw one the other day of a department that has a brand new bloodhound, and he's like 12 weeks old. And the cutest thing you've ever seen, like all he's tripping over his ears. And somehow Basset Hounds got in my algorithm in the last week, and I keep getting Basset Hound puppies, and their ears are like longer than their whole body, and they're stepping on them, and oh my gosh.
I saw a video once of this cop pretending to be a police dog to get teenagers out of the forest.
Oh, and barking at them. Yeah, it works.
And
It
they
works.
released them and they got caught.
The number of times that I've heard a canine officer say, "Come out or I release my dog," and then you see immediate action because this is not playing. My favorite one is still this one though, where it's a car chase. They crash into a telephone pole. He gets out and he starts running down a gravel driveway into the woods. Does anyone see this? The canine's dog's name is Shep because the guy releases him and he goes, "Get him, Shep!" And the dog just tears. And the whole time the guy is screaming, "Get him, Shep! Get him, Shep!" And the dog gets a hold of the man, and the man keeps running and he's dragging the dog. And the officer absolutely, "Get off my blippity blip dog. And then, I mean, it's a great one to watch. I love that. So, good. It's very good. It's very good. Now, one of the canines that Cole works with in Waco was actually shot recently, trying to get a suspect out who was hiding, and he's now decommissioned, but he survived. But they airlifted him. Canine dogs are treated like officers, right? So like the charges for hurting one are not quite equal to hurting a human, but they're pretty high up there, and they're given full honors when they die and all of that. So they do use bloodhounds anyway in police, and then they also use typically, what other two breeds are typically used by police? Do we know?
German Shepherd and Belgian Malinois.
Yeah, German Shepherd and Belgian Malinois. And Belgian Malinois are the freaking like parkour dogs of the planet. Like they're insane. They're insane. Huh.
You can take them skydiving.
Oh yeah, you can. All right, what are these? Does anybody know what these are, Barrett? Do you know?
The one right is a Dalmatian.
You got both of those. That's a Shiba Inu and a Dalmatian. So that's a Japanese dog there. Now, what group do these fall into, do you suppose? You know? Working dogs tend to be like your sled pulling dogs. So this is the spillover catch-all group. This is non-sporting. So these are the guys that aren't specifically bird dogs, they aren't specifically hounds, they aren't specifically sled pulling dogs or whatever. It's the overflow, it's the hodgepodge group. So you have things like Shar-Peis and Tibetan Spaniels and Tibetan Terriers and Chow Chows and things. Some of these are used for protection, like Chow Chows are protection dogs, for example.
What's the one in the bottom left, black one?
That is a Tibetan Terrier.
Oh,
They're cute. Oh, bottom left?
yeah,
Oh, a Skipperky.
yeah.
Yeah. And then the poodle next to him. All right, now how about these?
Cool.
One of these is pretty uncommon, but you might know it. Do you know, Caitlin?
Great Dane and a mop.
Great Dane and a mop. You got half of that right. The mop has a name. Do you know it?
Commodore.
Commodore. You know it. Okay, not too many people know that dog. I have yet to see one in real life, only in dog shows. And I watch the dogs. If you don't know, by the way, when the Macy's Thanksgiving parade ends, it's the Westminster Dog Show, and I prefer that to the Macy's Day Parade. I watch it every year.
I get to see it every time because it's always on the TV at my grandma's.
Yes, and it's great, right? Right. So that's where you see these guys.
I think it's doing the Super Bowl, but they have
The Puppy Bowl.
Yeah.
That's great fun. All right. So what group do you suppose these are in? Great Danes and Commodores. Do you know?
I'm gonna say big.
Big. So these go in your working group. So if you look in here, you're also going to find your Saint Bernards. You're going to find your Rottweilers and Bull Mastiffs. Like those are your defense dogs, right? Rottweilers, Bull Mastiffs, Mastiffs, English Mastiffs, Neapolitan. Anybody know Fang, Hagrid's dog? Neapolitan Mastiff, right? Your Great Danes, but also your sled dogs. You're going to find oh wait, no, not in this group. That's in the other group. So anyway, you'll find all these dudes in here, and they're pretty darn cool. There's some fun ones in there. All right, and then lastly, what are these things? Besides fluffy and kind of sort of cute. Do you know, Lucas?
One on the left. The Papillon, I think.
Not a Papillon, but that's a good guess.
I know it. I just don't.
Papillons look a lot like long-haired Chihuahuas, but they have.
That's not the Maltese, right?
It is a Maltese.
Oh.
Yeah. Do you know what the other one is?
No.
So these both are named for where they come from. We have a Maltese and a Pekingese. So the Maltese is from where? Malta. And where is the Pekingese from? It's in the first. Peking is right there. Chinese dog, right? Now there was a Pekingese that won Westminster for like a year or two in a row. You couldn't even see the freaking thing's face because like they put it on the ground. It looked like an alien because it was like all this just hair. You couldn't see its feet. And it just had this like tiny nose and eyes. You couldn't even, it just lumbered around. I was not impressed. Do you know a dachshund has never won Westminster? I'm still resentful of this.
Yesterday, a lady with a Maltese was like walking with her dog, and I had just gotten back from like dropping off a kid at their house. And it was snowing, and the lady yelled at me and said, "Merry Christmas, it's snowing!" And it just like started barking at me. And
It's not quite Christmas, but yeah, we got flurries. So what group are these little silly ones in? You know? Toys. Okay, so these are toys. Purpose of a toy. Be cute, sit in my lap. That's the purpose of a toy. These are companion dogs. So these dogs have a great temperament for being cuddly, friendly, except for maybe like this one right here is not necessarily known for being friendly. Which one is that? Chihuahuas and kind of monsters. You look at him, you're like, how cute. They'll take your fingers right off, right? All right, and then lastly, all right, what do we have here? Caitlin, what do we have?
Corgi
Mm-hmm.
and an Australian.
Corgi and an Australian Shepherd. That's in specifically is a Cardigan Welsh Corgi, not a Pembroke Corgi, and an Australian Shepherd with a beautiful merle coloring, right? And what group are these guys in? It's given away.
Have you ever seen
Oh.
mix between a corgi and a husky before?
Oh, that sounds awful.
Of course.
I hope the mother was the husky and not the other way around. What have you got? That's your herding group. Now, this is where you're going to find a lot of your smartest breeds because they have to strategize and think. So your Aussies and your Border Collies are some of your absolute smart. I had a dog in high school that was an Australian Shepherd Border Collie mix. She had like human level intelligence. I swear it was insane. Like my mother would let her out in the backyard and she'd look at her and she'd, cause Abby would go out and she'd just wander around the field behind our townhouse. And my mom would look at her and go, Abby, do not go left, do not go right, go straight. And she would go straight. Dog, like they're very smart.
In our neighborhood, there's an Australian Shepherd and Dachshund mix
Oh yeah.
named Finn, and he's like so wiggly.
Yeah. They've got mini Aussies now too that are very cute.
I
They're
have two Aussie.
fabulous.
Aussie.
I would have an Aussie again if they didn't shed so much. They would make my husband sick. So, oh yeah, they will.
They'll go.
My Aussie Border Collie cross could understand if she did something that it could elicit a laugh. So then she would do the behavior again to get the reaction. And they're very smart, like very smart. So anyway, these are your different breeds. Yep.
Fun fact, husky and a pug mix is called a hug.
Ew. Yeah.
So my bird uses her paws, and like when she like gets on her phone, she'll come up to you and she'll knock it out of your hand.
Yes. Yeah. Like pay attention, touch me. Yeah. No, my dog would like, she'd put her paw on your nose and then you would laugh and then she'd look at you and she'd wait till you stopped laughing and then she'd put it back. Like, okay, I'm doing this to make you laugh. So our most popular breeds by year, little Frenchies have been winning out for several years now. Now, the problem is with this, historically speaking, anytime a breed becomes really popular and say your average Frenchie is $600 to $800, I don't know what they sell for, but it's probably in that range for a Frenchie puppy. So somebody goes, I'm going to get on this bandwagon and make some money. And then they're going to breed irresponsibly and they're not going to breed them well. They get inbred. You end up with genetic issues because they're inbred, but that happens anytime a particular breed becomes more popular over others. So if you look in history, you can see different breeds and look at there, go figure that in the 60s or whatever, poodles were popular. They had just come off of people's skirts. So it was kind of a thing, right? And different breeds. Now, I will note to you that the German breeds like Dachshunds in particular and German Shepherds went hugely out of fashion during World War II because they were German, as if it's their fault, but they were like, oh, it's a Nazi dog. I won't have a wiener in my house. Are you kidding me? No, I mean, come on, people are dumb. But anyway, so golden retrievers for many years, they tend to stay in the top 5. So here are your breeds and how they rank right now. So like 1 through 50, we've got French Bulldogs, Labs, Golden Retrievers, German Shepherds, Poodles, Dachshunds, Beagles, Rottweilers, Bulldogs, German Short-Hair Pointer at number 10. Go figure. Yorkies, Aussies, Cavalier King Charles, Cane Corsos. If you've never seen a Cane Corso, they're big and looking like dogs. Pembroke Welsh, Corgis, Doberman, Pincers, Boxers, Miniature Schnauzers, Bernese Mountain Dogs at 19. Shih Tzus, Great Danes, Pomeranians, Boston Terriers, miniature American Shepherd. I'm not sure what that is. I know what the mini Aussie looks like, but Havanese, which is a really cool looking dog. Siberian Huskies, Chihuahuas, English Springer Spaniel, Shetland Sheepdog, Border Collie coming in at 30 because people aren't crazy. As awesome as they are, you better. That dog is a commitment. That's not just a dog. That is a daily commitment. Yeah, they're crazy. So anyway, going on and on and on, you see Brittany, Cocker Spaniel, Belgian Malinois, Basset Hound, Vizsla. Vizslas look like Weimaraners, but they're red. They're Hungarian. They're beautiful. English Cockers, Maltese, Pug, Collie, Mastiff, West Highland White, Shiba Inu, Rhodesian Ridgeback. You ever seen them? They look kind of like labs, but they're red, and the center line of hair along their spine goes the opposite direction from the rest. They're really cool. Papillons, Portuguese water dogs, which are like black small versions of the Commodore. They have the little mop coat too. Newfoundlands, Dalmatians, Australian cattle dogs, and Whippets, who apparently greyhounds and Whippets, which are like the fastest dogs, right? Like greyhounds can run flat out if you've ever seen them race. They are couch potatoes. They sit around and they will be lazy and they will do absolutely nothing. And then they're sight hounds. They'll see something and then they'll go. And suddenly they're lightning fast. So they go from like bro on the couch to track star in 30 seconds. They're pretty fascinating. So we have hybridization. So hybridization is what? What do you call hybrid? How do you define it? What's hybridization? Caitlin, what you got?
Crosses dissimilar individuals to bring together the both
Yeah.
parents and the offspring.
Yeah, so you're crossing dissimilar individuals to bring together some desired traits from both organisms. And this can be, hybrids are those organisms, and they can be dissimilar members of the same species, or in some cases, different species. Great example here. Oh, and it says they can often be hardier than the parents. And this is like mules is your great example. You cross a horse and a donkey, get a mule. Mules can be much hardier than the horses and the donkeys. They can do the pack work that the horse and the donkey can do. They're better on slopes. They're better on trail work and things like that. We took my mom horseback trail riding a couple years ago for a birthday gift, and they put me on a mule and not a horse. And I was like, oh, I was grateful, man. She was awesome. Like everybody else is bumping all around and I'm just lumbering up the hill on her. She was great. Well, then we have a zonkey, which is obviously a cross between a what? A zebra and a donkey. So what does that tell you about their chromosomal makeup?
They're special.
They're what? What?
They've got white and black stripe.
Right, but their chromosomes, what do you know if you can breed a zebra and a donkey?
Same number.
They have the same number and they align the same way. So here's a zonkey, in case you ever wondered. Isn't it cute?
Like a pig.
It's so cute. I like it. So that's how you get a zonkey. Yep. So you cross dissimilar individuals to bring together the best of both worlds. Another example is Brangus cattle. So this is a Brahma cow. They're from India, and they are really great at withstanding heat and harsh conditions because parts of India are really hot and dry. So they're very good for withstanding heat. And then Angus cows are known for what? Oh, the best steaks come from Angus cows, right? They've got really good meat, right? So if you cross an Angus with a Brahma, you get a Brangus, this dude over here, which means it can withstand the heat and still produce really delicious steaks. So, hey, now you can have steaks somewhere where there's hot weather or whatever. Texas, yeah. Brangus cattle have become quite popular. A lot of people will breed them. All right, what do you get when you cross a poodle and a golden retriever? Somebody, I'm sure, has one, or you know?
Labradoodle.
Not Labradoodle, but you're close. Golden doodle, you got it right. Yeah, you're on the right. Yeah, Labrador. The Labrador and the poodle are the Labradoodle. So why do we do that? Because now we get a hypoallergenic dog. Poodles are hypoallergenic, but standard poodles are big. They're tall, and you got to deal with their coats. You know, you got to go get them cut. Why do they cut them like that, by the way? Why do they have those little puffy things on their joints? Do you know why? It's to keep their joints warm. That's why poodles have like on their butt and then like on their knees and on their feet, they have the little puffs because they're trying to keep their joints warm. But then they cut the rest of their hair off because it's kind of a pain. Meaning it's less likely to trigger an allergy. So if you're allergic to dogs, poodles, Bedlington Terriers are the most hypoallergenic of dogs, by the way. And they look like tiny little lambs, if you've ever seen one. Their little faces are even look like lambs.
Mom had a Yorkie because she has a dog allergy. So we say if I get a dog, I'm getting a poodle or a poodle mix.
Yeah.
So that they don't shed.
Yes. Now, why do they pick a golden retriever? Because golden retrievers are known as lazy man's dog. I got that from a trainer who used that term, lazy man's dog. They want to please you. They want to make you happy. They're like everybody's favorite bro. Hey, what are we up for? Are we going for a walk? I'm up for a walk. Cool. Are we watching TV? I'm up for that too. What are we doing? Are we eating? We're not eating. Okay, I'm fine. They're like the chillest, happiest, make you happy dogs. So when you put your lazy man's dog with your hypoallergenic dog, you get this little fluff ball. Now there are some veterinarians who will tell you they aren't fond of these because they haven't gone through the whole, this is still new. Like we've only been doing this about 20 years. These little what they call the boutique breeds, the doodle breeds. And they can be a little behaviorally unpredictable, some veterinarians will say, because we don't know what to expect from them because we haven't necessarily like perfected that. It's not a new breed yet. We're just playing around. All right, and what do you get when you cross a tiger and a lion? What do we get?
Liger.
A liger. What are ligers known for? Oh, someone help me out. Someone tell me what ligers are known for. Drew? Their skills and magic. If you haven't seen Napoleon Dynamite, you are missing out. But his favorite mystical animal is a liger, a cross between a tiger and a lion. They're known for their skills and magic. They're not really obviously known for their skills and magic, but according to him, they were. Who has not seen this movie? Okay, assignment. Between now and next week, you have to watch this movie. You have to watch Napoleon Dynamite. Write it down. Napoleon Dynamite. It's amazing. It's weird. Yeah, it's weird. It's an indie film that became like a cult classic, and it's weird. It parts of it you're like, why are they doing this? This makes no sense, and it's just strange. But and they got paid almost nothing for it, and now it's huge.
I know a guy who, when he was younger, Julie Andrews came to his Barnes and Noble and signed books, and he was wearing a Napoleon Dynamite shirt. And I don't remember what it said, but it was something questionable. And Julie Andrews was like, "What's that from?" And he's like, "It's Napoleon Dynamite. You should totally watch it." He was like 8 years old.
She would like it.
Totally watch it.
Oh, she would like it. Yeah, she's cool, man. I love her. So anyway, you get a liger. So how do we increase our variations? Another way is biotechnology. So now we can skip all of the process of breeding, which obviously is complex, involves living animals, involves their behavior, or plants, involves behaviors where animals are concerned. Animal husbandry is like a whole field in and of itself where you're going to breed horses or cows or like the amount of effort that goes into breeding racehorses, by the way, is off the chart. It is ridiculous. There's so much money in it. They don't even like, a lot of the times they don't even let them breed. They will collect sperm from the horse and put it, the stallion, and put it in the mare because they don't want the mare to get injured. Like, because they can get injured because horses are big. So yeah. So we can introduce mutations on purpose, which can be good. Breeders can increase genetic variation within a population, introducing mutations, which are the ultimate source of biological diversity. Biotechnology is when you start applying technological processes and invention to this particular field. So we can manipulate the genetic makeup of an organism. And we selective breeding is one form would be like agriculture, but then you end up using chemicals and things like that in agriculture. So you can get in here and genetically modify your plants so that they don't require pesticides because they become pest resistant genetically. So, everybody automatically jumps on, a lot of people will jump on GM crops and GM vegetables and go, these are bad for you because they're genetically modified. Well, maybe they are, but that doesn't necessarily mean they're bad. And certainly you can increase your yield and decrease pesticides and other things by genetically engineering. So mutations are not always harmful. Y'all know that. An example would be oil-eating bacteria. We have produced bacteria that we can put where there's an oil spill and it will eat up the oil. That's pretty darn cool, right? There's the oil and the bacteria can eat that. They're small. Millions can be treated with radiation and chemicals at the same time that increases the chance of producing a useful mutant. And scientists have produced hundreds of useful bacteria. Some of them are going to consume oil from an oil spill. And working on bacteria can clean up radioactive substances, metal pollution. There's all kinds of things we can engineer our bacteria to do for us. Now, that is what a wild banana looks like. Do you want to eat that? No. Look at the seeds. That's wild, right? That's pretty wild. So polyploid plants or polyploid plants, you can do it either way, have 3 or more complete sets of chromosomes rather than the usual 2 found in your diploid plant. And drugs that prevent separation of chromosomes during meiosis can be useful in plant breeding. Yep, we can drug plants. And then what you get are cells that have many times the normal number of chromosomes, and then plants grow from these, and they're called polyploid because they have many sets. Instead of haploid or diploid, they're polyploid. And then in animals, though, this is usually fatal, but plants tolerate it better. So we can specifically breed our plants. And now we have like less seeds. This is what's going on here. So any banana you've ever eaten has been genetically modified to some degree. And you can quickly make new species. They can be stronger, larger. We do this with citrus fruits too. Now, inbreeding, y'all know what this is, right? This is continued breeding of individuals with similar characteristics. So yeah, these little guys here. So you cause some big problems with inbreeding. You need variety in your gene pool to have healthy offspring. This is why it's illegal to like marry your cousin or your brother or your whatever, because that's not good. Because then you end up not having enough genetic variation, and you have things like too many people in this family group carry the same recessive allele for this particular problem. And if you breed within that group, like, hemophilia and the royal family. Do you remember this from like a week ago? Right? We talked about this. This is how it happens, right? You get this overabundance of a particular disorder or even just like developmental problems, like embryological problems, if your genes aren't varied enough. So if you bring two recessive alleles together, obviously you can have a recessive trait that might not be a good one. But they do it sometimes with animals because they're trying to maintain this desired characteristic. So this is what I'm saying about Frenchies. If you're a not so reputable breeder and you're trying to get like that blue color Frenchie, it's so pretty. Oh, okay, I'm gonna breed a bunch of these blue Frenchies together to try and keep getting more blue Frenchies. Sooner or later, you're going to be breeding ones that are somewhat related to each other, and then you get problems. So it helps ensure that characteristics make each breed unique are preserved. Yeah, sure, but it can be very risky. And most members of a breed are genetically similar. And if you're too genetically similar, that's not great. It's going to increase the chance of crossing between two individuals that will bring together two recessive alleles for a genetic defect. Now, let's talk about dogs again. The one I know best, Doxies. All right, Dachshunds can have dapple markings, which in other breeds is called merle. Okay, so when we looked at that Australian Shepherd, it had that patchwork coat, was real pretty. It's called merle in a lot of breeds, but in Dachshunds, they call that dappling. When it's spotting and it can be beautiful. When one dapple dachshund is bred to another, you get a dog known as a double dapple. And the resultant dog is really pretty, but there are problems with double dapples. So the dapple or merle gene for dachshunds is dominant, and these combinations of genes can be as follows. If you have a heterozygous right there, you're going to see the dapple. So you have a big M, little m, because remember, it's the merle gene. These are beautiful, healthy dapple dachshunds in high demand. People love them. They're super cute, right? If you breed two with recessive alleles together, you're going to get a solid colored dog. So you can get a black and tan, or not solid, but not dappled dog. So you get a black and tan. That middle one is what they call wild boar, and that's typical on the wire hair dogs. And then you can get like a shaded red at the bottom. But if you breed two dapples to one another, you risk getting, think of your Punnett square, even if their makeup is this, big M, little m, you're going to get a big M in the mix, right? And you're going to get a double dapple. Now they can be really pretty. I mean, those are pretty. Look at that one at the top. It's got like big patches of color. They're beautiful dogs. But you can identify double dapple dachshunds easily because their patches are big and they tend to have a lot of white. Sometimes they'll have an entire white ear or an entire white face. It's not good because they can be born blind in one or both eyes. If there's a white patch, it's typically the eye covering the eye behind the white patch. So that little first dog risks that eye under the white patch being blind. They can be born with only one eye or no eyes or very small eyes. So it's interesting how eyes are linked to pigmentation. Remember that people with albinism can have vision problems? Again, that's not completely unrelated.
If you have the heterozygous one that's dapple, it's still dominant gene. So why is it not just completely? Why does it not do the same thing as this one?
Right. So it has to do with, again, we talked about like the turning on and off to produce the different colors during embryologic development. And for some reason, when you have the two dominant genes together, they don't even exactly know why, but when you like the mechanism, but the two dominant alleles together produce the double dapple, which comes with all the health problems. Whereas if you have a dominant allele and a recessive allele, you're fine. But it's just the two dominant alleles together that cause problems. Because remember, we talked about most of the genetic disorders are recessive, two recessive alleles, but you can have some where it's the two dominants. So they can be deaf in one or both ears. Some of these problems are lethal. They are already susceptible, dachshunds are already susceptible to certain health problems, and these get exacerbated with double dapple. So that includes things, typically their biggest risk for dachshunds is IVDD, but intervertebral disc disease. Their spines are so long, their discs get injured easily. That's why you don't drop them or let them jump off of high furniture, even though they will, because they are hell-bent on doing whatever they want. But yeah, so it can cause problems with their gait. They can have problems with their kneecaps becoming displaced, especially when they get older. And they can have very brittle bones and Cushing syndrome, which has to do with how their body's filtering out fluids and things of that sort. And then they can have thyroid problems. Now, if you unintentionally breed a double dapple, it's unintentionally, it's irresponsible, because you can genetic test your dogs with Embark or another program that'll tell you what you're breeding. So that's irresponsible. But if you do on purpose, it's disreputable. And I will tell you, the dachshund breeder community will shut you down if they find out you're breeding double dapples on purpose. Accidents happen, but you can prevent them because now you can genetically test every dog you breed. So if you're a reputable dog breeder, you've already done that. So now we talk about cloning. So this is when you create a genetically identical organism. And in 1952, we created our first clone, and it was a tadpole. That's not so hard to understand because frog eggs, as we've talked about before, develop externally. They're not inside of a mother. They're floating on the water, so they're easy to manipulate, right? So Scottish scientists cloned our first mammal, though. Does anybody know what the first mammal to be cloned was? Oh, Isabella.
Where?
Yes, it was Dolly. That's Dolly in the bottom there. The little one is Dolly. Isn't she cute?
Then how was the, oh, they named the one that was cloned, not the one cloned.
Yeah, no, she is the clone. She's the baby, right? That's Dolly. And that was 1996. So that means I was in college majoring in biology when this happened. So this was a big deal. It was a huge big deal. So much so that our April 1st edition of the student newspaper, which is always a spoof version, included a story about one of our students cloning another student, and I was the pregnant surrogate for that other student. And it got, the whole school was like, hey, what y'all been doing up there? I'm like, what happened? Oh, you haven't seen the paper yet, have you? A friend of mine wrote the article. It was quite amusing. So this is 1996. So Dolly suffered from lung cancer and arthritis, and she was put down by lethal injection in 2003. So she lived 7 years. And since we have cloned cows and pigs and mice and cats, and we've moved on. So how did we do it? Here's how we did it. And this is crazy. These Scottish scientists it's really smart. So they obtained an egg cell, in this case from that Scottish blackface sheep. Anybody seen Babe, Ba, Ram, You? Yeah, okay, anyway, they got the egg from her. And then they removed the egg's nucleus. So now it's just kind of, no genetic material because we took out the nucleus. Then they got a somatic cell or a body cell from a mother, in this case, that fin dorsate that you see up there towards the right. And it came from an udder. So they used an udder cell from this fin dorsate. And then they took that genetic information from that udder cell and put it into that empty egg cell. And then the third sheep came along, who was the surrogate ewe. They let it grow into an embryo a little bit, and then they implanted it into that surrogate down there. So which of these sheep is Dolly going to be genetically identical to? The fin dorsate at the top, because it's her genetic information that got put into the little kind of empty egg of the Scottish blackface, and then it was grown inside the surrogate ewe. So this is how it was done. Really smart, like it's creative. So it was genetically equal to the somatic cell donor, which is that fin dorsate, but there were issues. Animal clones in general show high rates of death, deformity, disability. Dolly was the only success out of 276 tries. That's stick-to-itiveness if I've ever heard it. Imagine doing that 276 times until you make it work. Oh. Now, is it natural? No, not really. It's not the way nature reproduces, right? So these are the questions that we ask ourselves about cloning. Is it natural? Would you want to be a clone? Would you want to be cloned? No, and maybe some of you are like, that could be fun if that person did like all the dirty work and I did the fun stuff, but yeah.
Isn't Tom Brady's like dog cloned or
Oh,
some
I don't know, could be.
some football players' dog.
I
was
wouldn't put
cloned
that
of
past
the
someone.
other dog.
Yeah, it's possible. I have no idea. Is it okay to clone human embryos for research and then destroy them later? These are the questions we get into. These are the questions that come up when you start talking about this stuff. How about this? What if you could clone your own cells or those of a family member to replace damaged organs? Is that different? Would that be okay? Would you do that? Right? There are all kinds of ethical questions that get involved when you start messing around with the way nature has always reproduced. There are some definite positive uses for this. There are some definite sticky gray areas. There are some definite, this is terrifying, we shouldn't do it things, right? So it's always lots to talk about. So benefits are things like this. If you improve it, you could reproduce animals with specific benefits. All right. So say you find cows that produce good lean meat or whatever, and you just want to keep producing that same cow. All right, that's cool. That's cool. And you could mass produce them. That could be great. That could be really cool. But you could also repopulate an endangered species. Now that could be a good use, right? That could be a very good use. What's the movie where they cloned an extinct species and then made a whole amusement park out of it, everybody? Oh, was that fun? Should we do that?
Yeah.
Oh, does that sound like a great time? We'll just do that. Why not? What's stopping us? You know, these are the questions to ask. You could produce whole human organs from just a couple of cells. That could be very useful for people who have kidney failure or heart failure or liver failure, right? I mean, you can see how this could be, or skin. Could regrow skin, you know, patches of your own skin to put on you if you get burned or injured or whatever. Stem cell research is used, can use cloned embryos. This is one where that kind of strides that line where people will go, we shouldn't clone humans. And we're like, well, what if we cloned an embryo? That way you're not having to harvest more embryos. So people get, like I'm saying, lots to think about. This gets very sticky, very, very sticky.
It kind of makes sense that Dolly was the only one that looked at like 300 because if you get the genes from an older sheep, it probably wouldn't.
And that's another question. That is definitely a question that people have thought about. Your cells and your whole body kind of is working on a timeline, right? Clock is ticking, clock is ticking. Does it matter how old the individual is that you harvest the clone cell, the cell to be cloned from? If it's a really old, if you get one of your grandpa's cells and clone it to make your kidney, is it only going to live however long your grandpa was going to live? Like, these are all still things we're still learning, we're still working our way through. There's things they can do genetic engineering wise, which is our next topic, that can kind of help work out some of those wrinkles and make things maybe develop differently than they otherwise would develop. So we can manipulate human, we as humans can manipulate an organism's DNA through the process of genetic engineering. So examples, one would be recombinant DNA, which we're going to talk about, cloning, which we just talked about, and transgenic organisms. All right, so let's talk a little bit about recombinant DNA. So this is when you get DNA from two sources and you can combine them together and you can change the genetic makeup of things. This can be really, really, really useful. So we cut DNA with what we call restriction enzymes, and then we introduce a new gene into that organism's existing DNA. So say you have a bad gene in your string, you can cut that out, put a good gene in its place. You can see how that could be good, right? How that could be a helpful thing for some people. So this is how they actually do it. A little graphic of how they do it.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as ECO-R1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of ECO-R1, for the base letter sequence GAATTC.
We found it.
The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, ECO-R1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture media. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So this is how we get things like certain drugs, for example. Bacteria produce those drugs for us because we've taken their circular prokaryotic DNA plasmid, taken a chunk out, putting what we want, and now that bacteria is going to produce a particular protein that we're going to use. So this is very common now. It's used quite a lot, and it's very, very helpful. So you see primarily in medicine and diagnostics. Great example, probably your best example, insulin. Oh, anybody know anybody who takes insulin for diabetes? Yeah, have an insulin pump or whatever. Well, that's how they're growing that insulin. They're using recombinant DNA technology on bacteria, and they can grow insulin, they can grow hormones, they can grow clotting factors, which is great for people with what Duspose disorder would need clotting factors. Yeah, there you go. Vaccines. It also facilitates disease prevention and treatment through diagnostic tests, experimental development of gene therapies. Very useful. Now, transgenic organisms. All right, what are transgenic organisms? So these possess a useful gene from another organism by way of recombinant DNA. So now we're not talking about little bacteria, we're talking about more complex organisms. So transgenic tobacco plant glows in the dark. What do you suppose, what gene or what organism do you suppose had the gene that they implanted into this particular, what's it, yeah, it's a firefly. Good, you got it right. That is a firefly. They took a gene from a firefly and stuck it in a tobacco plant. What's the purpose? We're going to make a whole field of glowing tobacco plants for people to go, ooh, cool. That's not the reason they do it, right? They do this because it's part of the process to learn how to do something functional and useful. So a lot of science looks really freaky and really ridiculous in its beginning stages when we're learning. And by the time we get to an end product, it's like, oh, of course, this is a logical purpose. There's a reason to do this. Otherwise, like, why would you make tobacco glow in the dark? That's just weird.
It could be for like a lamp.
Oh, a lamp? Yeah. Tobacco lamp in your house. Yeah, okay. That could be fun. So this is how your insulin is made for people right here. So bacteria produce important substances. So in order to get them to produce insulin or human growth hormone or clotting factor in large quantities, they're modified with recombinant plasmid right there. That's your insulin gene from a human cell stuck in a bacteria. You get your little transgenic bacteria and you get insulin in the end. Great. We need insulin. People need it. So that's awesome. But we can also use this to get better meat, resistance to disease, faster growing animals. Who likes a good pork chop? There you go. And we can do it with plants. And when we do it with plants, it is very important to our food supply because look at those percentages. 81% of your soybeans are transgenic, 64% of your cotton genetically modified. So when you're talking about soybeans, you may go, oh, I don't eat tofu. Look at any package. Like soy is in almost everything, man. It is everywhere. It is ubiquitous. Like it's in all sorts of things. And genes for disease resistance, or to produce their own insecticide, or heat resistance, or resistant to pesticides or weed killing herbicides, or to increase vitamin content. These are all things that we do with transgenic plants. So these little transgenic pigs are really cute. Oh, that's not so cute. Look at those.
Anti-aging therapy in mice, animals carrying human cells, breeding with helpless livestock, and building organs outside the body. Genetic engineers are constantly searching for answers to many social challenges. From global issues to medicine, these guys put everything to the test. One of the biggest goals of genetic modification is to reduce world hunger by producing more food. Featherless chickens have been genetically engineered to keep them cool.
Since I'm a geneticist, I was looking for a genetic solution. That means instead of putting the environment of chickens with feathers, is helping them feel cool by just removing the feather.
Without feathers and scales on their feet, these birds can grow comfortably in hot countries like Nigeria and Indonesia. The hope is that once successfully bred, the healthy commercial-sized birds will help feed underdeveloped countries. To fight global warming, some turn to invasive pigs. The industrial amount of waste from pigs is a serious environmental pollutant.
Pig manure has a very high level of phosphorus within it from normal diets to normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
But the remarkable thing is that the EnviroPigs carry a man-made gene. One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other from mice produces a genetic switch that allows this special enzyme to be made in saliva. These two pieces of DNA were combined to form a new compound gene that makes their poo more environmentally friendly. Medical researchers across the world are also turning toward transgenics for answers.
Like the word transgene, it means that the gene was transferred, foreign gene was transferred again.
Having successfully used transgenics to insert a jellyfish gene into a rabbit, when charged with blue ultraviolet light, the skin and the eyes of these rabbits glow green. But why make animals that glow in the dark? The incandescent gene allows researchers to easily track the movement of cells. By tagging certain cells with the glowing jellyfish genes, scientists can track which cells go where after an organ transplant. The technology is helping to find treatment for problems like blindness and bone disease. Glow-in-the-dark rabbits are just a start to the medical challenge that genetic engineers have taken on. No one said it was easy being green.
So those are actually really creative uses of technology, making pig manure less toxic to the environment, making rabbits glow so you can track the way cells develop. So for organ transplants, that's helpful. So in agriculture and industry, everything we eat and much of what we wear is going to come from living organisms, and the use of genetic engineering to try to improve products and plants is very, very common. As you can see, 93% of the corn produced in the United States is genetically modified. There have been over 20 years of research on GMOs that concludes that they're not toxic, so says the research. And studies have shown that GMOs have no impact on fertility or reproduction, and genetically modified crops can be resistant to certain viruses and yield more from less land. Now, that's a good thing. If you can get more crop out of less land, then that's less land you have to designate for farming. So you can see how that could be a very good thing. If you can get more meat out of a cow, then you can have fewer cows, then you can have less methane and less cow manure and less cow pollution, right? So ideally, genetic modification could lead to better, less expensive, more nutritious food, as well as less harmful manufacturing processes, ideally. We've had GM crops, or genetically modified crops, since the 90s. In 2007, 92% of your soybeans, 86% of your cotton, and 80% of your corn was genetically modified. And we're using things like bacterial genes that produce this as botulinum toxin. Harmless to humans in this case, the way it's being, no, it's not botulinum, BT toxin. Sorry, it's different, not botulinum. But anyway, it converts BT toxin into a form that kills an insect. So now we've got no pesticides needed. You can grow a whole field of a particular crop and not need to pour chemicals on it, chemical pesticides on it, because you've genetically modified the crop to begin with. And they can be resistant to herbicides. So you can spray your herbicide and kill all your weeds, but not harm your plants. And they can be resistant to viral infections and produce food that's resistant to rot and spoilage, which, as you can imagine why that's a good thing, and produce plastics for the manufacturing industry. So then we have our genetically modified animals. So cows, 30% of milk comes from cows with hormones made by recombinant DNA techniques to increase milk production. So, you know, industrial dairy farms are nothing like, you know, your little storybooks as a kid where the farmer goes out with a little bucket and does this. It's not that. Like, I don't know if you've ever watched videos of industrial dairy farms. It's wild. Like, it is a process. They bring all these cows in, they have these giant things like hook up, they flash fire under their udders first to like flash kill any bacteria that are there, and then they hook them up to the milking machines. It's pretty wild, actually. So pigs can produce more or lean meat at higher levels and more omega-3s. Salmon have a growth hormone that make them grow more quickly in captivity. Now, this is the fun one. We found a way to put spider genes into goats so that we can manufacture silk from goat milk. I'm not kidding. And it can be made into light, tough, flexible material, military uniforms, medical sutures, tennis rackets. Human genes plus goat milk give you antibacterial goat milk. And you can save endangered species doing this kind of stuff. And in 2008, the US government approved the sale of meat and milk from cloned animals. Did you know that? Ah, yes. In case you didn't know. Now, in case you didn't believe me about the goats and the silk, she's gonna prove me right.
I'm Danielle Gassabide. I am a master's student at Utah State University, and I work in Dr. Randy Lewis's spider silk lab. We are trying to produce the proteins that spiders use to spin all of their fibers. This is the spider that we base all of our genetic sequences off of. It's called the Netheloclavipes. It's also called the banana spider. You can't farm spiders, they're cannibalistic, and you cannot get actual protein from them. So what we're trying to do is produce all those types of silks in other hosts. Those hosts include alfalfa, bacteria, we use E. coli, goats, and silkworms. The goats are not affected adversely. They are genetically modified from the time of conception, basically. So they're modified in the embryo. And then everything after that just comes from breeding. So when we breed the goats, not every baby has the gene. The only difference in the goats is they produce a lot of proteins in their milk. These goats are special because they produce one extra protein basically in their milk. So yeah, if you tasted it, you look at it, you look at them, they look exactly the same. Spider silk is biocompatible, which is one of its really cool properties. So potentially we could use these products for medical uses and replace things that are used now, make them better, make them stronger, make them easier on your body. We've also had interest in the textile industry and in adhesives, making eco-friendly adhesives that don't have harmful byproducts, that aren't adhesives that aren't petroleum based. That's kind of the great thing about this research, is the kind of opportunities are endless. Because it has the potential to do so much good, it would be a shame to not harness the power of genetics.
Anybody ever had a suture kind of work its way out of your body instead of stay put? Every time I get stitches, if they give the nylon ones, my body's like, this doesn't belong, and it pushes it out. But like spider silk is biocompatible. So you could potentially make sutures, for example, that your body's not going to reject or push out. You could make other things too, using that same protein from goat milk. Who knew that? Isn't that weird? Now, biologic drugs are a whole different class of drug, and these are typically injectable. They're also extraordinarily expensive because a lot of research goes into them and they're difficult to make. But this will show you how they're made. Ooh. Oh, that's scary. It's a scary movie. Ah! Okay. Ooh, okay. I don't know what that's about, but that looks interesting.
Biologic medicines are proteins produced inside living cells, which are mini factories generating the genetically engineered protein treatments. It's a different approach from traditional drugs, which are manufactured using chemical synthesis. Biologics offer hope where other therapies have failed. To mass produce these biologic medicines, scientists use the molecular machinery inside living cells. The process starts with the insertion of genetic material into Chinese hamster ovarian cells. They're known as CHO cells. They keep them frozen in liquid nitrogen until the next step. In the seed lab, a technician thaws the cells in warm water. He transfers them to a glass flask that contains growth media. The growth media is comprised of hundreds of nutrients, including vitamins and minerals. It's a recipe to help the cells multiply. A worker transfers the flask that contains the cells to the production side of the plant. The team injects the mixture into a 5-gallon capacity bioreactor. Inside, a propeller agitates the mixture to stimulate growth. Over a 3-week period, the number of cells increases exponentially. Each new cell is programmed to produce the target biologic protein. Next, a technician measures salts and other dry ingredients. He's preparing a recipe for purifying the protein liquid once it's been separated from the cells. He labels the bag and records the information separately, as required by government regulations. He adds the ingredients to purified water in a mixing tank. An automated system keeps tabs on the condition of the purification solution. After separating cellular material from the protein mixture, the team pumps it through steel and glass columns. Here, the purification solution works with resin beads to separate the protein from the impurities. And finally, this technician takes a sample and conducts a test to ensure the medicine is sterile. Grown in living cells, these biologic medicines are now on their way to potentially making a difference in people's lives.
So a lot of these are going to come in their own injectors, little pens, injectable pens. So you may be familiar with some of them. Like, here are some examples. You've probably seen the commercials for these, right? These look familiar? Yeah. Notice that a lot of them end in UMAB or IMAB. That's like the suffix for this particular category of biologic drug, but they end in UMAB or IMAB. A lot of them do. Like, aren't they fun? Look at that one. Avastin, bevacizumab. Humira is adalimumab. And if you listen to the commercials, they say it at the end, and they always say it really fast, and it sounds like a different language because it's so crazy. So these tend to come in like self-injector pens. A lot of them do. It's kind of like an EpiPen sort of a deal, but you push the little, you don't have to slam it, just push the little button. I take one for my lupus. I take Taltz, which isn't on here. You just saw the process for making them. I'll let you just guess. Without insurance, which thank God I have, how much do you think my once a month injectable biologic costs per dose? What do you think?
Are we saying with or without insurance?
Without, what would it be? Do you know?
I'm gonna say $4,000 without 2,000 with.
Okay.
7,000.
7?
10,000.
None of you have gotten close yet.
13,000.
A little more. It's 10,000. Yeah. $10,000 roughly per dose. Now it's like 110 with insurance, and then there's like a special government program that gets it to you for free. I don't know. But yeah, they're expensive. These are very expensive. So you don't just walk in and get them. You have to get approval for this, and it's a pain in the butt process to get them. But they're so effective that they're turning them. Now this is the drug. They are turning out biologics for everything now.
There's insulin pens are like $2,000 a pen.
Yeah. And those aren't even this category. So yeah, you know what I mean. So yeah, these things they're very expensive, but they alter how your body's functioning in a way that's different from the way medications of the past have. And they're extremely effective. Mine's night and day. Like, I would not be able to stand and talk this long. I would be laying down by now without it. So I'm grateful for it. So early physicians extracted substances from plants and animals to cure patients, right? Those are what our earliest doctors did. Oh, we'll take bark from this or this flower and we'll make a tea and it'll make you better. And that's what we did. So in the 20th century, when vaccinations came in, we've saved countless lives through vaccinations. We had Jonas Salk showing up and all these people. And then now we have recombinant DNA technology, which is some of the most important advances in prevention and treatment of disease. So we've come a long way. So goals to prevent health problems, infant blindness, for example, is one of the things they're working to prevent. Transgenic plants and animals to make human antibodies. Future transgenic animals may supply us with human proteins used in disease prevention. And we can simulate human diseases caused by defective genes, which means then we can use those simulated diseases within animals to trial various drugs and treatments on them, right? So those are models that are based on simulations, follow onset, progression of disease, and test new drugs. So you can create lab organisms, animals that have a disease. You can genetically modify the organism to have this disease, and then you can track its sort of its natural course. And then you can also try your different drugs out on it and see what works. And Alzheimer's and arthritis are great examples of things that they're making models for. They're making model animals for Alzheimer's and arthritis. Recombinant DNA technology made important proteins that could prolong and save human lives, and human growth hormone is used to treat pituitary dwarfism. It's not widely available because it's mass produced in recombinant DNA, recombinant bacteria. Human growth hormone, like some people have used it like to jack themselves up, but that's not the purpose. Like it's actually given to people who are lacking in certain natural endocrine processes that would make them grow.
Do you get taller though?
You can. I do have a friend whose son went on it because he was like 15 and he was still, oh gosh, maybe 65 pounds or something. He was very small. He wasn't growing properly. It was an endocrine problem. So they replaced the hormones that he's missing. So that, and one of them was human growth hormone so that he could grow properly. And then we've got insulin for treating diabetes, blood clotting factors for hemophilia. So gene therapy. Now, this is the process of changing a gene to treat a medical problem or a disorder or disease. And we can take absent or faulty genes and replace them with normal genes. So it can allow the body to make the proteins that are needed. We can engineer viruses so they can't reproduce. We can put DNA with therapeutic genes into modified viruses. We can infect patient cells with a virus because in theory, the virus is going to insert this new healthy gene into the targeted cells. Remember how viruses are going to inject their genetic material into a cell so that it makes more of itself? If you make a virus so that it is not harmful, but it is able to produce a specific protein based on its DNA profile, then you can give a human that virus will inject into the human cells, and now the human is making the protein that they were lacking to begin with. So there's all kinds of good options for this, but some of it is very high risk, some of it is questionable, some of it is still on the cutting edge. So all cells in the human body have your identical genetic material, almost all, technically not, but same genes are not active in every cell. So we can use DNA microarray technology to study hundreds or thousands of genes at once and understand the levels of gene activity, because the genes that produce your hair are, you know, the cells that produce your hair obviously don't have the genes turned on that produce liver cells, right? Like the cells that produce your skin don't have the genes turned on that produce liver or kidney or something else. So we can actually take this technology to kind of understand outside of the body how all of those things are working. And now we have genetic testing available for hundreds of disorders. So, and then there's this. We did this a couple weeks ago. What's this thing called? Remember what that's called? Do you remember?
It was like DNA
electrophoresis was the process, right? And it produces this DNA fingerprint. And we know that no two human beings are genetically exactly alike aside from identical twins. So DNA fingerprinting analyzes all that DNA so that you can compare one individual to another and you can identify people. So we used to have to require, and this is the craziest thing, we used to have to require hair with a root or a decent sized blood stain or a decent amount of saliva or a good sperm sample or any of these things from a crime scene or whatever. Or, you know, if you had you need a good size sample is the point. And you could, like, somebody had a coffee cup and you swab that coffee cup, you basically destroy what sample you had in testing it because we needed so much of it. Right now, if I did this, I just left skin cells right here, and this is touch DNA. That's all they need now. That's all they need. It's crazy. You are dropping your genetic material everywhere all the time.
Aren't they using that for like the Louvre? I don't know how to pronounce it. Museum.
The Louvre? Yeah, I think so. With the trying to find out who did what recently. Yeah, I think they might be. But touch DNA, I mean, they would prefer a good bodily fluid sample or something like that, but they can use touch DNA.
Lots of people have already been identified and like they've gotten really far into the investigation, but they haven't found any of the stolen items yet. And it's probably never going to be seen again as they're breaking down into their raw materials.
It's pretty crazy that I don't know if anybody's ever seen The Great Muppet Caper, but I was instantly reminded of those scenes where they're breaking in to try to get that diamond or whatever it is. That's about the Louvre thing. That was my mind. Oh, they caught him red-handed. What color are his hands now? That whole bit. Yeah, so now we don't even need a big sample anymore. We can do this with very small samples. And they would prefer a big sample. It's easier, it's less expensive. But we've been doing this kind of technology since the 1980s. And forensics, obviously, you know what that is. This is the first DNA fingerprinting case.
Body of a 15-year-old girl, Linda Mann, is found in a village near Leicester.
It's still raw. And I can't believe that after 35 years. She's such a lovely girl. It was wrong.
3 years later, and a second girl is found less than a mile away. Dawn Ashworth was also 15.
When Dawn's body was found, police knew straight away they were looking for a serial killer. What happened during the course of this investigation would change police detective work around the world forever.
David, just explain what all this part of stuff is. These are mementos from the case, are they?
David Baker, who's now 84, was leading the double murder investigation. He'd arrested a 17-year-old, Richard Buckland. Buckland had admitted the second murder, but there was a problem.
There was no circumstantial evidence against him on the first murder, and he totally denied it.
You were convinced it was one murderer?
Yes.
David had read in a local paper about research being done at the University of Leicester. Professor Alec Jeffreys had attached DNA from cells to photographic film. The result, each person's individual DNA could be read. David Baker contacted Professor Jeffreys to get Buckland's DNA tested.
Nobody had ever attempted a crime scene DNA analysis, a real-life crime scene DNA analysis before.
Would the test find the killer? Would Buckland's DNA match that found at the crime scene?
They came back not connected. The result was stunning.
A blow to you?
Well, it was a blow, because it meant that we'd got the wrong man.
Buckland was released. The very first time DNA profiling was used in a criminal case, it exonerated a suspect. David Baker was back to square one. 2 murders, 1 killer, no suspect.
It says, will the killer strike again? How can you help to trap him?
We made a decision there and then to use the DNA system as part and parcel of the inquiry.
What happened next was extraordinary. Police asked all the men who lived in the local area to give a sample of their DNA. More than 5,000 men gave a blood sample voluntarily, and eventually the killer was caught.
This is Pitchfork. He went from being a very normal family man to a murderer in a split second.
Colin Pitchfork, a local baker, was sentenced to life, the first person ever to be convicted on the basis of genetic evidence.
Today, there are ongoing debates about DNA, how the information is stored, who should have access to it, how it's used. But Linda's family have no doubts about genetic profiling and the historic role it played in solving a double murder.
I find it amusing. His name is Pitchfork. Just, yeah.
Apparently he was like, he gave it and he was bragging that, I heard that he was bragging that I'm the killer and nobody's gonna find me, and that's how they found
I think that's right. Yeah, there's a lot of cases like that. And then if you want to know about confessions, like that guy confessed, they said, if you want to know about the ultimate in false confessions, if you're interested in that kind of thing, you need to look up Henry Lee Lucas. Because dude, I don't know if does anybody know him? Do you know him? Dude got arrested, and he did kill a couple people. But within a matter of a few years, he had confessed to like 600 murders. And basically, like, the Texas Rangers had him, and they would like treat him like royalty. Like, they'd bring him into this interview room every day. And investigators from all over the country would come with their unsolved cases, and they would bring him cigarettes and strawberry milkshakes because that's what he liked. And they would just, here are your cigarettes and strawberry milkshakes. Tell me about, do you know about this girl? Let me slide this picture of her across the table. She was murdered in her garage. How'd you do it? Oh, I found her in her garage and I killed her. Oh, well, he knew she was in the garage. She must have done it. Like literally, that's what it was. It got to the point where it was the DA in Waco who was like, "This makes no sense." Like he was supposed to be killing somebody in Seattle at 4 o'clock on Friday, but on Saturday at noon he was in Fort Lauderdale, Florida. Like he can't have done this. He can't have done it. But it's so now we try and back up confessions with forensic evidence because we know better.
When did the murder happen?
This one, 1983.
On the Fairbrook podcast,
Yeah.
for, like the season 2, that guy who was probably coerced into a confession.
Yes.
I was like,
Yes.
about to crash out. I was like,
Yes. It's crash out worthy. So your genome is your complete set of genes located in any one of your cells, right? And we have our Human Genome Project, and it has done all these wonderful things for us. It has helped us with everything from just understanding how things work to improving diagnosis and pharmacogenics and all this good stuff. Ethical questions, like we said. Possible outcome of the Human Genome Project: no more genetically imperfect people, like Stephen Hawking. Oh, well, no more depressed people, like Vincent van Gogh. Okay, well, no more physically challenged people, like FDR. Okay, well, no more blind people, like Stevie Wonder. Okay, well, no more people with multiple handicaps, like Helen Keller. So obviously, ethical concerns, right? Now, yes.
My brother is convinced Helen Keller doesn't exist.
Yeah.
He's like, there's no way that's a real person.
This is I'm not even going to preface this. I'm just going to let you watch this. I'm going to tell you what you're watching because it's unreal.
Mammoth meatball.
Is one mammoth meatball. And by mammoth, we don't just mean big. Food scientists in Australia are bringing back a meat not seen in thousands of years, the woolly mammoth.
Because this protein is literally 4,000 years old, we haven't seen it for a very, very long time.
A company called Vow injected mammoth DNA into the muscle cells of a sheep. For the missing strands of genetic code, the scientists added African elephant DNA because they're the mammoth's closest non-extinct relative. The combination was grown in a lab, resulting in 14 ounces of meat. Apparently, the combination has a distinct odor.
It's super fascinating to think that adding the protein from an animal that went extinct 4,000 years ago gave it totally unique and new aroma. Something we haven't smelled as a population for a very long time.
Mammoth meat won't be on sale anytime soon, or possibly ever. The creators say they haven't even tasted it. Who knows what kind of allergens could have existed thousands of years ago? Instead, this experiment was to generate buzz around the possible environmental benefits of creating meat instead of raising it.
We wanted to get people excited about the future of food being different to potentially what we had before. But there are things that are unique and better that don't know that are necessarily needed.
For Inside Edition Digital.
All right, now the question is, who would try the mammoth meat? I think I would try it. Just to see it, but that's wild. This like real life Jurassic Park, but instead of for an amusement park, it's let's go with that spaghetti. Mammoth spaghetti. I wonder what the Italian word for mammoth is. Mammoth jerky. Well, this isn't as far off as you might think. Maybe not mammoth meat, but wouldn't it be great if you didn't have to farm a bunch of cows to get beef? You could just grow beef. Oh!
I'm Brian, and this is IGN.
Check out their balls.
Things will be eating their 3D printed meatballs in the not so distant future.
This guy's funny, actually.
Space 10, IKEA's Green Living Design Lab is looking for tomorrow's meatball, a healthy and sustainable meatball that'll satiate our unsustainable appetite for meat. According to Space 10, we have an increasing demand for food. That demand is creating serious problems for our planet. It's impacting global warming, wasting water, destroying forests, and causing soil erosion. The United Nations predicts that our food demand will increase by 70% within the next 35 years. So we need to be smarter and more efficient about the way we produce our food. It's a really smart paragraph for the way I started the story talking about balls. This means being open to eating stuff we haven't considered before, like artificial meatballs made with algae, bugs, and beet leaves. Hey, it's better than the horses they use now. This isn't the first time we've seen 3D printed food. IGN staff ate 3D printed pizza at CES 2015, and we reported that German candy company Catchies is 3D printing gum candy in some of their cafes. Would you eat a 3D printed meatball made from insect protein? What's wrong with you? Let us know.
What's wrong with you?
6.
It's a bit of a thing. Here, I'll pop these up so you can see these answers. Whoop. Sorry. I mean, that's, if you think about it though, like cattle farming in particular is causes huge environmental concerns. And if there was a way to grow like the healthiest possible beef without involving all those environmental concerns, like that can be appealing to some people.
It would be really funny if Big Flavor did that.
Yeah, I don't think they're going to be the ones. Somehow I kind of doubt it will be them, but still.
It's funny. This big would be funny.
Okay, hold on, because I still have a couple more slides here. Hold on. Making sure you see these answers. And yes, they are in Dropbox. They're already there. I moved them there this morning.
You're my favorite.
Oh, gee, thank you. Don't tell your mother that. And then you have a test coming up, and these are your topics. Oh, go back. Oops, sorry. I bumped it. I bumped the button and it went away. There it is. I did it again. I'm too fast. Sorry. There it is. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Operons is my last one. Don't over click. There. There you go.
My mom said she's a few chores for me to do.
She's a what?
My mom says she has a few chores for me to do.
A few chores for you to do? Yeah, uh-huh. That's what it looks like? Yeah. You've completed that. All right, let me see. All right, check this over. Let's see how you did.
Let's see. That's good. I don't get
Y'all have a great week.